Hello students, welcome to the Bionurturing channel. In this video, I am explaining about plasma membrane and their models and basics. What is the plasma membrane? Plasma membrane is a outermost layer of the animal cell, and it is a boundary between the inner environment of the cell and the outer environment of the cell. Okay. So this is the cell. This is inner side of the cell and this is outer side of the cell. So this layer which is separating the inner side and outer side of the cell. Okay, this layer is known as a plasma membrane. Okay, it is present in animal cell and plant cell, both cells. Okay, then basic importance of the plasma membrane. Okay, this is the structure of the plasma membrane. Okay, whenever you observe under electron microscope, it will look like this. Okay, this is the structure of the plasma membrane. Okay, and it was made up of three components. Number one, cholesterol. Number two, phospholipid. Number three, proteins. Okay, so this blue color structure is known as the proteins and the purple color structures is known as the phospholipids, and in between them their small structure is known as the cholesterol. Okay, this is the structure of the plasma membrane, and the functions of plasma membrane is, first one is cell security gate. Okay, it is the outermost layer of the animal cell and it doesn't outermost layer of the plant cell, because plant cells already have cell wall. So cell wall is the outermost layer of the plant cell. When it comes to animal cell, cell wall is absent. Okay, so plasma membrane is the outermost layer of the cell. Okay, that's why it's known as a cell security gate. Okay, whenever whatever enter into the cell, it need to be passed through the plasma membrane. It need to be passed through the plasma membrane. Okay, it need to pass from the plasma membrane. That's why I'm saying it is a cell security gate. And the second function is selectively permeable. Okay, it allows only particular molecules. Whenever it doesn't allow any unwanted molecule for the cell, it allows only particular molecule which is useful for the cell, like water, oxygen and nutrients. Whenever the components which was harmful to the cell, it doesn't allow. Okay, from the outside or inside, whatever, outside or inside, it will control the momentum of molecules. That's why it's known as a selectively permeable membrane. It is selecting the molecules and it is allowing the molecules. That's why it's known as a selectively permeable membrane. Then super thin and flexible. It's the thickness of the plasma membrane. The thickness of the plasma membrane is 7.5 to 10 thickness. Okay, and it is a dynamic. Why I'm saying it is dynamic means dynamic. It contains several components like proteins, phospholipids, cholesterol or carbohydrates. There are several type of molecules, not same. It is present different structures, like protein is different structure and lipids are different structure, and it doesn't static. Okay, all the phospholipids and proteins which was present inside the plasma membrane, they will move from one place to the another place. It doesn't stick on a particular place. Okay, it will move everywhere, like phospholipids move like this, and proteins also flip their places. It was moving, it is moving everywhere, all the molecules will be moved. So that's why it's known as a dynamic. And elastic, why it was moving means there is a fluidity. That's why without elasticness, it can't move one place to the another place. So it is a dynamic and elastic. And then microscope view, we can see only under electron microscope. Like plasma membrane we can't see with our eyes. Generally cell is a microscopic structure. Okay, plasma membrane is a one of the part of the cell. So that's why we can't see with our naked eyes. We can see under only microscope. Okay. And then chemical composition of plasma membrane. Number one is lipids, number two is proteins, and number three is carbohydrates. Okay. Number one, lipids. Okay. Lipids are the main important composition of the plasma membrane. Okay. It contains more composition comparing than the proteins. Okay. So lipids is a mainly made up of with phospholipids. Mostly lipids are made up of with phospholipids, and it contains two parts. Okay, like this, head and tail. Okay, so this is hydrophilic and this is hydrophobic. So hydrophilic means the head of the lipid which loves water. It will attract to the water. It will interact with the water. But the tail which was present, it doesn't interact with the water. It hates the water. It doesn't react with water. That's known as a hydrophobic. Hydrophilic means which molecule which loves the water, that is known as a hydrophilic. The molecules which hate the water is known as a hydrophobic water, and it is also known as a amphipathic in nature. Why I am saying it's amphipathic? Amphipathic means any molecule which contains two type of natures, that is known as a amphipathic. Like in this phospholipid we can see two type of natures, hydrophobic and hydrophilic, two type of natures. That's why this lipid is known as a amphipathic in nature. Okay. Then second one is a protein. Okay, proteins are the major important structures in plasma membrane. It involves in the cell signaling and transportation, everything. So here proteins are two types in plasma membrane. Number one is integral protein, and number two is a peripheral proteins. I will explain you what is integral protein, what is peripheral proteins. Okay, so this is the integral protein which transferring the two layers of lipid bilayer. Okay, whenever the protein was transferring two layers of the lipid bilayer, that is known as a integral protein. The protein which was transferring two layers of the lipid is known as a integral protein. The protein which was present only one layer of lipid bilayer is known as a peripheral protein. It may be outside of the cell or inside of the cell. Outside of the cell means outer side of the plasma membrane, or inside inner side of the cytosolic side of the plasma membrane. It will may be present in outside or inner side of peripheral proteins. Only one layer, it was attached to the one layer of the lipid layer, so that is known as a peripheral proteins. Okay, then carbohydrates. So carbohydrates, it will attach to the proteins here. Like will explain you. Generally it was present outside or inside. Okay, I mean it is maybe a cytosolic side or outer membrane side. Cytosolic side or outer side. Okay, like this. Like this. It may be linked with the phospholipids or maybe proteins. Okay, that is known as the carbohydrates. Okay, whenever it linked with lipids, that is known as the glycolipids. Whenever it linked with proteins, that is known as the glycoproteins. Okay, it was very important for the signaling. These carbohydrates are very important for the signaling. These are the composition of plasma membrane, and then models of plasma membrane. Okay, models of plasma membrane means it was all the models which was explaining the plasma membrane. Okay, these are all models which is explaining structure of the plasma membrane. It contains several models. Okay, there are several models which was explaining the plasma membrane. Okay, plasma membrane structure. These all models are involving for the discovery of the plasma membrane structure. Okay, so we can know through the periodic way. The first one is lipid bilayer model. Okay, it was proposed by Gorter and Grendel. Gorter and Grendel. It is also known as a Gorter and Grendel model. Okay, it was proposed by Gorter and Grendel at 1925. Which year? 1925. And who was proposed? Gorter and Grendel. Okay, so this experiment was done on the red blood cells, RBC, and they give some statement. They give some statement like they are saying lipid lipid is not a single layer, it is a double layer. They are saying it is a double layer, it's not single layer. Okay, it is arranged in a bimolecular layer, lipid bilayer, not only single layer, two layers. They are saying lipid present in two layers, that is known as a lipid bilayer. Okay, and they explain structure of the plasma membrane like this. So this is the one layer and this is the one layer together known as a bilayer. Okay, so when you observe there is no proteins here. So when it comes to the structure of this lipid bilayer, so each lipid molecule contains two parts. Number one is head and number two is tail. Head is a hydrophilic head and tail is a hydrophobic. Hydrophilic means it loves water, it will interact with the water. Okay, and hydrophobic means it hates the water. Okay, so why they assumed this is a correct model? Because at the time they think like that this water, these heads are water loving, so that's why they think like that. These heads was helping the water through the inside of the cell and doing all the physiological functions like that. Okay, they assumed like that at the time. That's why they believed this is the this is the structure of the plasma membrane. But it doesn't accept it because they have some drawbacks. Okay, demerits are there. Okay, the demerit is mainly there, there is no any protein here in this model. There is no any protein. They are explaining only about the lipid bilayer. So how some molecules doesn't enter into the this uh, these proteins. Okay, so some molecules it will enter through the proteins only. So some molecules doesn't enter through the lipid bilayer. Okay, so like lipid soluble proteins, so lipid soluble proteins will be entered through the phospholipid layer. But what about non like protein soluble proteins? It can't enter because they didn't mention the any protein layer here. Okay, so how can I say lipid soluble proteins? So I will explain you clearly. Lipid soluble proteins will go through the phospholipid bilayer. Okay, suppose this is lipid soluble protein. Okay, it will allow through the plasma membrane because it is a lipids. Here lipids is present. But suppose protein soluble molecule is there. This is protein soluble molecule. It doesn't enter into the plasma membrane because there is no any protein here. If there is protein, through that protein it will enter into the cell. So here it was failed because there is no proteins. Number one drawback, and number two is it doesn't explain the transportation. membrane proteins. It doesn't explain the transportation of through the membrane proteins. So it failed here. This is the major drawbacks of lipid bilayer model. Okay. Then sandwich model. Sandwich model also known as a Davison and Donnelly model. Okay, it is also known as a Davison and Donnelly model. It was proposed by the Davison and Donnelly. Okay, like Hugh Davison and James Donnelly. These are the two persons. It was discovered at 1935. You need to remember the scientist names and the years which was they proposed. That is important, and which statement they give and what are the major drawbacks of this model. These are the key points you need to remember and noted. Okay, here sandwich model also known as a Davison Donnelly model, and it was proposed by Hugh Davison and Jameson Donnelly at 1935. Okay, 1935. So here they was explaining the structure of the plasma membrane like this. Okay, it is phospholipid bilayer. This is a phospholipid bilayer. Okay, it was sandwiched between the two protein layers. So can you imagine why it was named as a sandwich model? Okay, everyone know what is sandwich. We know sandwich. We ate lot of sandwich, chicken sandwich, veg sandwich, like that. Okay, so you can imagine like a sandwich. This model, imagine like a sandwich. Okay, this outer layer is a bread. This inner layer also bread. In between the two breads, this stuff is known as a phospholipid. So this phospholipid bilayer is known as a stuff between the two protein layers. So phospholipid bilayer was sandwiched by two protein layers, under two protein layers. Okay, so that is known as it, named as a sandwich model. Because why they put name, to understand very well in the model of the sandwich. Everyone knows sandwich, so that's why they put the name and explain through the sandwich type. Okay, how can it was clearly mentioning phospholipid bilayer is sandwiched between the two proteins. They are explaining protein layer, lipid bilayer, and again protein layer. This is a sandwich. Lipid bilayer is sandwiched between the two protein layers. Okay, then they tell another thing in this model. There is a tiny pores. Okay, there is a tiny pores here and here. So layer for water movement, they're simply saying, okay, they're simply saying there is a tiny pores. Through that pores, the water movement will be done from inside the cell to outside the cell. Okay, through these pores, small pores, the transportation will be done. They explain like this, but the main drawback is it doesn't explain the lipid soluble substance passing. Like I clearly mentioned you, I already told you in previous model also. So in this model, they said through the small pores the molecules will be entering into the cell and outside of the cell. But okay, protein soluble, but suppose this is a protein soluble molecule, it will enter like this. In this place it will stop. Why? Because here there is no protein. This is a phospholipid bilayer. Okay, it can't enter into the phospholipid bilayer. It can enter only until the protein layer. Okay, but suppose lipid soluble protein, this is a lipid soluble protein, it doesn't enter into the first layer also, because this is a protein layer. So here this is a protein layer. Lipid soluble molecules or substance, it doesn't enter into the membrane. Okay, but suppose this is a protein soluble molecule, it enter allow only particular layer, it doesn't enter into the bilipid layer of the plasma membrane. Okay, so it doesn't explain the transportation of the substance, like lipid soluble substance and protein soluble substance. Both type of substance, they didn't cross this plasma membrane. they didn't cross. So if in case substance doesn't cross or from outside to inside, how cell will be transport the molecules? How cell will be survive? So this is the question. This is the major drawback of this sandwich model. I'm clearly saying again without any confusion. But suppose there is a this is the phosphate soluble protein soluble molecule. Okay, it need to be enter into the cell. Okay, it will enter until this point only. It can't enter into the phospholipids. Why? It is a protein soluble molecule. Okay, and another one is a lipid soluble molecule. Okay, it doesn't enter into the first layer also because it contains first layer contains protein, so it doesn't allow, it can't pass. So here it was failing the explaining of the transportation of the molecules or substance from inside or outside of the cell. Okay, this is the major drawback of sandwich model. Okay students, in this video I explain only two plasma membrane models. The remaining models will explain in part B. Please must watch part B also to understand well. If you have any doubt, comment it down. For more videos, subscribe to our channel.